For the nasal representation is inhibition of hypopharyngeal squamous cell carcinoma migration by a proteolysis targeting chimera of histone deacetylase 8, presented by Phurinut Somchai from Faculty of Medicine Ramathibodi Hospital, Mahidol University.
Good morning, everyone. My name is Phurinut Somchai from Faculty of Medicine Ramathibodi Hospital, and today I'll be presenting my research about the inhibition of hypopharyngeal squamous cell carcinoma migration by a proteolysis targeting chimera of histone deacetylase 8. Hypopharyngeal squamous cell carcinoma, or HSCC, is a malignant tumor arise from the hypopharynx and can extend into the oropharynx and esophageal inlet, and it is a member of a group of cancer called head and neck squamous cell carcinoma. The HSCC has the worst prognosis among the other cancer in its group because patients are usually asymptomatic and leading to a delayed diagnosis. So a metastasis, especially to the lung, contributes to mortality in patients with HSCC, as illustrated here. The current treatment, including a primary surgery and a chemoradiotherapy. In chemoradiotherapy, a cisplatin will be used as a chemotherapeutic agent, but it has numerous side effects, including allergic reaction, gastrointestinal disorders such as severe vomiting, and severe kidney problems. And this may make the patient cannot be able to tolerate it. The histone deacetylases, or HDACs, are enzymes that play a key role in epigenetic regulation. Many studies show that the activation of HDAC8 isoform stimulates cancer progression by various mechanisms, such as enhancing cell proliferation, suppressing apoptosis, and triggering metastasis. The overexpression of HDAC is found in many cancers, including oral squamous cell carcinoma, which is also a member of head and neck squamous cell carcinoma. So a proteolysis targeting chimera, or a PROTAC, it is a technology that induces endogenous protein degradation through a ubiquitination and proteasomal degradation. This makes it has a significant higher advantages over conventional protein inhibitors because it can suppress all functions of the target of interest. In this case, our target protein is HDAC, and it not only has the catalytic function, but it also has a scaffolding function and interacts with transcription factors. Therefore, inhibition of catalytic function alone may not be able to stop HDAC. So as I mentioned before, the HDAC8 is overexpressed in oral squamous cell carcinoma. Therefore, we hypothesize that the HDAC8 level, if we degrade the HDAC, we can inhibit the progress of HSCC. So we wanted to compare the effects of 5 compounds on cell proliferation and migration on a hypopharyngeal squamous cell carcinoma, or a FADU cell. Those 5 compounds include our compound, HDAC8 PROTAC. And since HDAC8 PROTAC consists of the molecules of HDAC8 inhibitor linked to a pomalidomide, we add those 2 compounds into our control group. We also add vorinostat, which is the pan-HDAC inhibitor, and a cisplatin, which is the conventional treatment. For the cell proliferation assay, we stain the cell with a dye called the deep red. We plate it, treat it, and then we trypsinize and analyze the mean fluorescent intensity, or the MFI. So the concept of this is every time the cell divided, the dye needs to be diluted into the daughter cell. So a low MFI will indicate a high cell proliferation, vice versa. So here's the result. As you can see here, the HDAC8 PROTAC at 2.5 micromolar has a higher MFI when compared to a control group and the other treatments. And it's also show a dose-dependent manner because when we increase the dose, the MFI also increases. We also determine the proliferation marker, which are Ki67 and PCNA using a real-time PCR. So the results show that the expression of Ki67 decreased nearly 2.5 times in a cell treated with HDAC8 PROTAC, and it decreased the PCNA expression by nearly 3 times. But as you can see here, the HDAC8 inhibitor alone does not show any effect on those proliferation markers. So it may suggest that the active site or the catalytic function that this substance inhibit may not be associated with the proliferation of the cell. We also determine the migration using a scratch wound assay. So what we do is we plate the cell and then we create a scratch wound using a special tools, and then we treat the cell and then we place it in a machine called a live cell analysis system, where there will be a microscopic camera capturing an images from the same field of view every 12 hours, and then we measure the distance between the wound and then analyze it. So this is how we do it. This is the special tool, and this is how the picture looked like under the microscope. So the result here, as you can see here at time 0, all wounds are equally scratched by the machine. And as you can see here at the yellow line, and we designated the initial wound width as 100%. And as the time goes, cells started to proliferate and migrate until the wound is fully closed, as you can see here in the control group. But at 2.5 micromolar, the HDAC8 PROTAC wound width at 72 hours time point is still remain at around 60%, and vorinostat is around 45%, while the other treatments are fully closed at around 60 to 72%. And if we do a statistic analysis, the HDAC8 PROTAC did not show any significant differences between its initial wound width and the current wound width. So the current means the 22 time point. And this slide shows that the anti-migratory effects of the HDAC8 PROTAC is dose-dependent, because as we increase the dose, the wound gap closes slower. But if you take a look here at the concentration of 5 and 10 micromolar, you can see that the wound width exceeds 100%. So that means the wound width is wider. It may suggest that at higher concentration, my substance, the HDAC8 PROTAC, may have the cytotoxic effect on the cell. So the conclusion of this is the inhibition of the cell migration of HDAC8 PROTAC occurs at the same concentration at 2.5 micromolar. And it also reduces the cell proliferation. And all the effects are greater than the vorinostat, which is the pan-HDAC inhibitor, and it's the first FDA-approved HDAC8 inhibitors as well. So these results highlight the very potential that HDAC8 PROTAC is a very promising candidate for preventing a metastasis in hypopharyngeal squamous cell carcinoma. And I'd like to thank you, my team, and my institute for the continuous support, and that will be all for my presentation. Thank you.
So, thank you for your excellent presentation. My question is about what about the stability of the HXP PROTAC, right? And because you are going to use it for the reduce the proliferation of the cancer. So, if you want to invest into the in vivo, what about the stability?
Can you repeat the question again? Sorry.
Because now you use is a in vitro work, right? But when you want to work with the in vitro, like in a mice model, how can you induce this with this model?
So, if I understand it right, you want to ask about the how to stabilize my molecules, right? To be able to use it in a in vitro study. So I need to tell you that this is the part of the very big project, which is a collaboration between two universities. So at that point, we need to communicate with the team, and that is not our plan just yet. Our plan right now is we wanted to confirm the anti-migratory and the pathway of it first before stabilizing it. Yes, we're planning on doing a transwell migration assay and a Western blot to determine the mechanism behind its effects. Yes. So the answer is we're not really planning about doing it in an in vitro study just yet.
From your result, it seems like your HDAC PROTAC have fairly the role to inhibit proliferation, right, in the earlier result. But here, in the wound healing assay, so to distinguish the proliferation and here, is it the possibility that the effect that you didn't see any migration here is derived from proliferation inhibition of your HDAC PROTAC?
So you're asking if it's whether this effect that the wound gets closer can caused by the differentiation of the proliferation of the cell.
Yes.
Right. Okay. So at this point, I think that we can differentiate it by the wound width because we know the rate of the proliferation. If you can see here, it proliferates and you can see that it closes pretty quick and also migrate, right? So, but when but if we inhibit the migration, it can still proliferate, but only the yellow line just moves just a little, but not as quick as this one.
Do you have any suggestions to investigate or test directly that your HDAC PROTAC is the effect on migrations without any interference or co-founding of proliferation?
Thank you for your suggestion, and that will be taken into my further study.
I think it's about the composition of your medium. So how many % of the serum that you add in the experiment?
This one, right?
Yeah, for the migration study.
Yeah, we use a 40% as our serum.
4%?
40.
40.
Serum.
I mean the fetal bovine serum you
oh,
added
it's
in the
10%.
medium.
Sorry, 10% of fetal bovine serum.
So if you add 10%, we cannot divide which one is migration or proliferation. So maybe you need to consider the concentration of the serum.
Thank you for your suggestion. Thank you very much, everyone.